Plants are described as being annual, biannual, or perennial. This describes how long it takes for them to complete their life cycle. Annual plants are given one reproductive event that occurs within a year. This could be a single season or over the duration of an entire 12-month period. The diagram here shows us summer annuals that begin to grow in spring, mature in the summer, and reproduce at the start of fall, and compare that to winter annuals who begin to sprout in the fall, mature in the spring, and in the late spring have their reproductive events. But all of this is occurring within a single year cycle. Biannual plants take 2 years, hence the bi, like bicycle. In this case, the first year is dedicated towards individual growth, self-sufficiency, and maturation. The second year marks the beginning of the preparation for reproduction and the reproductive events. Perennial plants are those that can take more than 3 years or that can persist beyond a single reproductive event. In this case, these plants will still die, but death is due to infection or trauma, not necessarily the completion of a life cycle and journey. So we can see in this diagram here, the perennial spent the first year growing, the second year maturing, and it reproduced in the late summer, early fall, and then it continued to grow to have a second reproductive event in the third year, same timing, end of summer, beginning of fall. And so we can see a large number of the plants that we know and depend upon for food fall under these categories. Annuals are those that have a single growing season. A lot of the vegetables that we eat, the regular produce. A lot of our seasoning herbs or roots take 2 years and are biannual plants. And then there are some that you plant once and you'll continue to get flushes of growth annually. These are the perennials. In order for flowering to occur, 3 developments must take place. The plant must meet sexual maturity. Its apical meristem has to shift from a vegetative growing meristem to a floral meristem. So this involves gene expression, changing the primary directive or goal of the apical meristem. And then it takes time for the plant to listen to those new instructions and grow the individual flower organs. All of this is controlled by genes. The plant has a set threshold in size. So the shoot, the stem structure will grow to a set size. And when it reaches that capacity, that triggers one of the signals to switch to a reproductive growth mindset versus a vegetative growth mindset. Then there's also the internal and external cues from the environment. External cues would be temperature, duration of sunlight. The internal cues will be the hormones that are being produced as well as the nutrient availability. Growing a flower takes only 3 genes. And this is an interesting model of gene efficiency. If you had 1 gene for each body part, that would take up a lot of our DNA. And so oftentimes what we see is that there is 1 gene, 1 protein. But depending upon which genes are expressed simultaneously, the size or functionality of that protein might be different. And this is best described by the ABC model in plants. In this case, there are 3 genes, gene A, B, and C, and they direct the formation of 4 floral organs. So they're guiding, building 4 body parts using only 3 bits of information. And they're able to do this based upon what genes are expressed individually and what genes are expressed in tandem simultaneously. The diagrams here have tried to be color coordinated. If gene A is expressed alone, we get a sepal. But if gene A and B are expressed simultaneously, yellow and blue make green, so we see the petal forming. Or if you're looking at the diagram on the right, I've got blue and yellow stripes to show that the expression is happening together. If B is expressed with C, then we get a stamen, as blue and red make purple. And then if C is ever expressed just by itself, we get the female carp. Gene efficiency is consistent with our 1 gene, 1 protein hypothesis. but it leaves room for differential protein expression depending upon simultaneous gene expression. The sepal is not a true leaf, but these are the protective leaflets that cover the petals to ensure that they're safe. The petals, what will exhibit color, scent, sometimes sugar tracks, honey guides, and they are meant to be attractions for the pollinator of choice. And then there's the stamen versus the carpel, the male versus the female bits. If a flower has all 4 parts, they are considered complete. If a flower has both the male and female parts, they are considered perfect, just like our funguses. Pollination is the process of moving pollen from the stamen to the carpel. And often one can tell just by looking at the flower whether it is a cross-pollinator involving 2 different plants or a self-pollinator involving the same plant. There are some physical differences because form dictates function. Be aware of these different features and how they might help a plant breed with itself or encourage a pollination event. If you were an outbreeder, you are trying to be flashy. You are trying to catch the attention of your pollinator of choice, which means you need many and large, brightly colored flowers to basically act as neon flashing signs to get notice. Those flowers should be scented and have nectaries to entice the animal in. And you would want to have the male parts very far from the female parts to discourage your own self-pollination. Meanwhile, the inbreeder can get away with having just a few flowers because all it would take is one reproductive event and they're good. Those flowers are going to be small because that's going to put the male parts closer to the female parts. Those flowers are probably going to be dull or monocolored, not bright. They'll have no scent, no honey, no nectaries. Why bother? They're not trying to impress anybody, and they don't need as many pollen grains because they're not taking a chance on another organism. They're just dropping one pollen grain or a few pollen grains down into the female bits, and that should be sufficient. These features make sense with the end goal. What are the benefits to being an inbreeder? Well, the positives are that it preserves a genotype, genes and traits that are well adapted to this environment. It also ensures that the plant will seed and reproduce without relying upon anyone else who may be fickle or absent. It also means that one reproductive event is possible, and they don't have to do this multiple times in the same season, hoping one thing sticks. However, the downside is a big downside. Should the environment ever change, and that's a big if, but a likely if, their old traits may no longer be suitable in this new environment. And so the diagram that I have here is showing black mice that are well suited to a dormant volcano, well suited to hiding out there. As they reproduce, they're going to preserve a well-adapted genotype, black fur, in this black environment. But should the environment change, climate change, and suddenly it starts snowing on this mountaintop, they are no longer suitable, lacking any light-colored genes that might help them camouflage in this new snowy environment. Those black mice stick out like a sore thumb and would be the first to go by any flying predators. What about the outbreeders? What are their pros and cons? The pro here is a big one. Increased genetic variability. Each reproductive event is a grab bag. Who knows what will happen? The reward of potentially being better than the generation before outweighs the risk. There is still a chance they might get all the cruddy genes and be slightly worse than before, but often, due to survival of the fittest, the ones that do survive are going to be better adapted. The con of an outbreeder is they are wholly dependent upon their pollinator. And that means what happens if your pollinator doesn't show? What happens if your pollinator becomes extinct? What happens if that year there is no rain or wind or other natural event that helps in your dispersal? So oftentimes we see plants have a dual strategy. Their priority is to outbreed, and they will have structures in place or strategies in place to avoid self-pollination, but they might have a backup plan. that in a pinch will allow them to do so if needed. So let's talk about these mechanisms to separate the gametes, either temporally through time or spatially through logistical space. One strategy is to temporally use time to separate the male and female parts. Know the difference between protogeny and protandry. Protogeny occurs when the female parts, the stigmas, become receptive before the anthers release their pollen. The female parts are open and ready to receive pollen grains, but the pollen grains must come from another plant, as that particular flower on that tree will not be shedding pollen at this time. So protogeny occurs in perfect plants that have both male and female parts, but because the females mature a whole day earlier, they will be pollinated by some other genetic information elsewhere, not by their parent tree. Protandry is the opposite of this. In this case, the males are receptive before the female parts. So again, we still see perfect plants. Protandry is more common than protogeny. Now, how can you recognize protogeny versus protandry? I like to look at the suffixes. The ogeny looks like an OBGYN, the doctors that deliver babies, whereas protandry, we can think of the androgynous, very manly features. Protandry, also, as a side note, is most common in insect-pollinated plants. The idea is the insect will arrive, pick up male bits from the first flower, and then when they move to the next flower the next day, they will be moving that information over to the then receptive females. Another strategy is to separate the male and female parts through space. In this case, you could have dioecious plants, imperfect flowers that are either all male or all female. This is a highly evolved trait, and it's very common in oceanic islands that use wind pollination to move them. The idea here is you want that wind to carry it farther away to other plants of a different gender. In this case, there is no backup plant. They must cross-pollinate or die. You could also have monacy. In this case, you have a single plant, but that plant has a variety of imperfect flowers, meaning there are some all-male flowers and some all-female flowers. This is also very common in wind-pollinated plants, as the idea is the wind will most likely carry that pollen farther away to a different plant to encourage outbreeding, but in a pinch, it will lead to some occurrence of self-pollination. And so monacy also has a backup plan available to it. These backup plans make sense as plants need to pollinate, especially those annual plants or biannual plants. They die at the end of their growth season. For them to reconstitute the next season over is not them, but their offspring growing in their place. Pollination is essential here, and therefore many species utilize methods that would promote cross-pollination first, but then allow for self-fertilization in a pinch. Here's an example of protandry. The male bits are ready first. The pollen is being shed. The hope is that an insect will land and take that pollen to another flower. Then the stigmas become receptive, and if they do not receive pollen from a subsequent visit by the insect, the stigma's petals will dip down and eventually touch the male bits below them. Only as a final resort, this flower would self-pollinate. Outbreeding is the ideal. So how can plants encourage outbreeding? The flower itself, the tree, is nothing without its pollinator. This is a story of coevolution, the simultaneous evolution of two organisms to become better and better suited for each other. Pollinators come in a wide variety of forms and shapes, and pollination can happen through wind, water transfer, but mostly it involves insects. And among all the insects, bees pollinate 65% of all plant life. To ensure pollination, plants employ one of two strategies. Either they can trick, but more commonly, they bribe them. Plants will evolve to be more and more effective at attracting their pollinator of choice, of deterring other species to decrease competition. Because it's not just about having a reproductive event, it's about having a successful reproductive event with energy efficiency. And the pollinators will get more and more effective at exploiting the resources offered, and that typically means taking more pollen grains with them. So as a result, you can often just look at a plant and know who's pollinating it. There are some key patterns that we'll get to. Bees pollinate 65% of all plant life. They're the most important group of flower pollinators, and they specifically see yellow and blue colors really well. And so most yellow and blue flowers are pollinated by bees because they are visible cues to them. Bee-pollinated flowers typically also have honey guides because bees are so dependent upon pollen for protein and nectar for their carbohydrates that this helps guide them to the nectar and brings them deep, deep down into the flower to where they get covered in those pollen grains. There will be scent for bee-pollinated flowers. It is one of the things that attracts the bees to that location. Birds have a good sense of color and are very attracted to red or yellow. These two colors also have the greatest contrast to the green foliage of the leaves, so it helps a bird spot them as they're flying overhead. However, birds have beaks, not noses, and so they do not have a keen sense of smell. So their flowers have little or no odor, as they're not necessary. The flower petals will also tend to be very small because they don't have to support the weight of the organism. They don't have to act as a landing pad because the birds can fly in their own right. Bats, being active at night, are going to be looking for white pollinated flowers that bloom in the evening. These flowers will have scent. They often have strong scents. But the idea here is that white against black has the greatest contrast and will stand out and reflect back the moonlight to help the nocturnal mammals find their home. Bats have long, sticky tongues and they eat a lot of the pollen. And so flowers that are specially adapted to bat pollinators, they don't just have sticky pollen that gets placed on the bat's face when the bat takes a snack. What happens here is the flower will almost explosively release pollen. It doesn't only coat the bat's face, it'll coat their back, their ears, places that their tongue cannot reach. And the idea there is that when it goes to the next plant, it will have missed some of the pollen grains further behind, and those will be the ones that participate in fertilization. After pollination, something occurs known as double fertilization. Plants are the only organisms in which this happens. Everything else, fertilization occurs using a single sperm and a single egg. But in plants, there are two sperm involved and one egg. The sperm is coming in through the pollen grains. The egg remains within the carpel. And the idea here is two sperm plus one egg creates a 3N, 3 sets of genetic information, nucleus. The first sperm and egg will develop into the future plant. But that second sperm will become the endosperm. It will ensure the nourishment of the plant during its time underground until it can reach the soil surface and begin photosynthesizing on its own. If a flower is fertilized, if it is pollinated, that flower will grow into a fruit. Fruit are described as coming in 3 forms: simple, aggregate, or compound. And this has to do with where and how many the ovaries are. A simple fruit, a good example of this is tomatoes, avocado, pear, or apple, is a single flower, and within that single flower, a single ovary. An aggregate fruit is where you've got a single flower, but there are many ovaries within this flower. A good example of this is raspberry or strawberry. And then there's the compound fruit, in which case there are many flowers, and within these many flowers are ovaries, and ultimately they will all fuse together to form one giant fruit. A good example of this is durian or pineapple. And this can explain why half of the pineapple might be really, really sweet and another half might be really, really sour, because there are different genetics coming into play at each of these ovary instances. Maturation is a series of stages that prepares a seed for germination. The first thing that has to happen is we have to break dormancy. Dormancy is a state of hibernation, if you will. It's a state of inactive limbo. Dormancy is not the absence of life, but it is a signal to hold in the current state. It buys the new seedling time. It protects it until conditions are right. And therefore, very specific cues are required to break this message, because following this instruction set increases the chance that the seed will germinate in a place away from the parent and at a time, the right season, that will be most advantageous to the new plant, to limit competition and to ensure ample resources to thrive. The primary cue for germination, to break dormancy, is the uptake of water. By absorbing water, the seed will swell, and this breaks the seed coat. This creates a gap now in which other cues can diffuse or osmosis in. This also allows the radicals, which are primitive roots, to move downward and start transforming into the root structure. Other important cues for germination is oxygen amount, the overall temperature, and the balance between light and dark periods. In place of pollination, what are some methods humans employ to artificially propagate plants? One of the things we can do is called grafting. It's used to produce novel varieties, novel meaning new, to encourage sexual reproduction between plants and the sharing and genetic recombination of their traits. Grafting usually involves two plant species. They're going to be closely related. And the idea here is a branch from one plant is inserted into a tree of another closely related species. So the two sets of the vascular tissues join together, and one root network will typically supply two trees. One is growing out of the other. And what this does is this puts the new genetic material in very close proximity to your other species, and this encourages genetic diversity. Grafting is very common in apple orchards, and it might be one of the reasons why we currently have 600 species of apples today. Cutting is another method that we can artificially propagate plants. It is the most commonly used method, especially if it's a woody or ornamental plant. The idea here is you cut a bit of the stem. You want to avoid any material with a flower bud because all the energy is then going to the reproductive bits, and it won't stimulate rooting if it's growing in the other direction. And then you want to minimize the number of leaves, because for every leaf you have, that's energy that you are expending to keep them alive. And what you want to do is expend energy to grow down. You want about 1/3 or 1/2 of their length to be submerged under soil. This will block out light cues and signal to the plant that it needs to turn on new gene expression to adapt its structure to be a root. And there are also chemicals and hormones you can coat them with to help signal this change. The last method is known as layering. This is a type of organized suckering. So we talked about stolons and rhizomes as mechanisms that the plant puts forth to send its shoots and create additional trees. Layering allows us to initiate this with branches in a similar fashion. And so the idea here is you could have a long branch that's growing at the base of the plant. If you bend it low, that flexible stem can be inserted below the ground. This breaks that 180 line. That sharp bend will often induce rooting. And then after a time, you can cut that part to separate the plant. We would cut here, and this would be its own root. And the plant will, a secondary plant shoot will come here, and we would have two separate plants. They would be genetic clones. But it's a quick way of propagating plants for commercial application rapidly.